Hello, welcome to Biology, the scientific study of life. Sequences related to transposable elements. Multiple copies of transposable elements and sequences related to them are scattered throughout eukaryotic genomes. A single unit is usually hundreds to thousands of base pair long, and dispersed copies are similar but usually not identical to each other. Some of these are transposable elements that move using enzymes encoded either by themselves or by other transposable elements. And some are related sequences that have lose the ability to move altogether. Transposable elements and related sequences make up 25 to 50% of most mammalian genomes, and even higher percentages in amphibians and many plants. In humans and other primates, a large portion of transposable elements related DNA consists of a family of similar sequences called allo-elements. These sequences alone account for approximately 10% of the human genome. Allo-elements are about 300 nucleotides long, much shorter than most functional transposable elements, and they do not code for any protein. However, many allo-elements are transcribed into RNA molecules. Their cellular function, if any, is unknown. Although many transposable elements encode proteins, these proteins do not carry out normal cellular functions. These elements often are described as non-coding DNA along with other repetitive sequences. Other repetitive DNA, including simple sequences DNA. Repetitive DNA that is not related to transposable elements probably arose by mistakes that occurred during DNA replication or recombination. It accounts for about 15% of the human genome. About a third of this 5 of the human genome consists of a large segment duplications, in which a long stretch of DNA between 10,000 and 300,000 nucleotide pair seems to have been copied from one chromosomal location to another on the same or a different chromosome. In contrast to single duplications of long sequences, simple sequence DNA contains many copies of tandemly repeated short sequences, as in the following example. Notice that DNA strand only GTTA, CGGTA, CGTTA, CGTTA, etc. In this case, the repeated unit consists of 5 nucleotides. GTTAC repeated units often contain fewer than 15 nucleotides, but they may include as many as 500 nucleotides. The number of repeated units at a particular site in the genome varies as well. For example, there could be as many as several hundred thousand repetitions of the GTTAC unit at one site. Altogether, simple sequence DNA makes up 300 of the human genome. The nucleotide composition of simple sequence DNA is often different enough from the rest of the cell's DNA to have an intrinsically different density. If genomic DNA is cut into pieces and centrifuged at high speed, segments of different density migrate to different positions in the centrifuge tube. Repetitive DNA isolated in this way was originally called satellite DNA because it appears as a satellite band in the centrifuge tube. Notice that. Separated from the rest of the DNA. Now the term is often used interchangeable with simple sequence DNA. Much of genome's simple sequence DNA is located at chromosomal telomeres and centromeres, suggesting that this DNA plays a structural role in the chromosomes. The DNA at centromeres is essential for the separation of chromatids in cell division. Centromere DNA, along with simple sequence DNA located elsewhere, also may help organize the chromatin within the interphase nucleus. The simple sequence DNA located at telomeres at the tips of chromosomes prevents genes from being lost as the DNA shortens with each round of replication. Telomeric DNA also binds proteins that protect the ends of the chromosome from degradation and from joining to other chromosomes. Genes and multi-gene families. We finish our discussion of the various types of DNA sequences in eukaryotic genomes with a closer look at genes. Recall that sequences coding for proteins and structural RNAs compose a mere 1.5% of the human genome. If we notice this figure, if we include introns and regulatory sequences associated with genes, the total amounts of gene-related DNA coding and non-coding constitute about 25% of the human genome. As in prokaryotes, most eukaryotic genes are present as unique sequences with only one copy per haploid set of chromosomes, but in the human genome, such solitary genes make up only about half of the total coding DNA. The rest occurs in multi-gene families, collections of identical or very similar genes. Some multi-gene families consist of identical DNA sequences, usually clustered tandemly, with the notable exception of the genes for histone proteins. Multi-gene families of identical genes code for RNA products. An example is the family of identical sequences encoding the three largest ribosomal rRNA molecules. These rRNA molecules are encoded in a single transcription unit that is repeated tandemly hundreds to thousands of times in one or several clusters in the genome of a multicellular eukaryotic. The many copies of this rRNA transcription unit helps the helps cells to quickly make the millions of ribosomes needed for active protein synthesis. The primary transcripts is cleaved to yield the three rRNA molecules. There are then combined with proteins and one other kind of rRNA, 5S rRNA, to form ribosomal subunits. The classic complex or the classic examples of multi-genes families of non-identical genes are two related families of genes that encode globins. Group of proteins that include the A and beta, alpha and beta polypeptide subunits of hemoglobin. One family located on chromosome 16 in humans encodes various forms of alpha globin, and the other one chromosome 11 encodes form of beta globin. The different forms of each globin subunits are expressed at different times in development, allowing hemoglobin to function effectively in the changing environment of the developing animal. In humans, for example, the embryonic and fetal forms of hemoglobin have a higher affinity for oxygen than the adult forms, ensuring the efficient transfer of oxygen from mother to developing fetus, also found in the globin. Fetus we also found in the globin gene family clusters and several pseudogenes. Non-functional nucleotide sequences quite similar to the functional genes. The arrangement of genes in gene families has provided insight into the evolution of genomes, we will consider some of the processes that have shaped the genome of different species over evolutionary time in the next session. Okay, notice also gene families. Notice here the human alpha and beta globin gene families and part of a ribosomal RNA gene family. These are three of the hundreds of the copies of RNA transcription units in notice also here RNA transcript. Okay, thanks for listening.